Welcome back everyone to another ACE lecture. In today's lecture we are going to discuss the nervous system. So basically the job of the nervous system is to monitor the body and to maintain its homeostasis by responding to fluctuations in the internal and the external environment. And this job is being accomplished by different divisions of the nervous system. So the two main branches of the nervous system are the central nervous system and the peripheral nervous system. The central nervous system further divides into two other branches, which includes the brain and the spinal cord. The peripheral nervous system, on the other hand, is divided into the sensory and motor pathways. The other name for sensory pathway is afferent pathway or afferent neurons, while the other name for motor pathway is efferent or effector neurons. Now the motor pathway itself is divided into somatic nervous system, like for instance our skeletal muscles that are under our voluntary control, and the autonomic nervous system, which is not under our conscious control, and these include the sympathetic nervous system and the parasympathetic nervous system, and we will discuss these two autonomic systems in further details. But sympathetic nervous system is also known as fight or flight response, while the parasympathetic nervous system is known as rest or digest response. Next, moving on to different cell types in the nervous system. So the nervous system has two cell types. One is neurons, which are basically specialized cells that respond to various physical as well as chemical stimuli in order to conduct electrochemical signals and release neurotransmitters. And there are three main types of neurons. Number one is sensory neurons, or the other name for which is afferent neurons. And these neurons will gather the information and transmit them to the central nervous system. Next we have the interneurons, which are located in the central nervous system, and they function as a link between sensory and motor neurons. So they are the link between sensory and motor neurons. And then finally the last type is the effector or efferent or motor neurons, the function of which is to relay information to the effector tissues, like such as muscles and glands. So it will affect muscles and glands and other tissues. Alright, now the second type of cells in the nervous system are glial cells, and basically the function of glial cells is to support neurons. They will provide supporting framework, they would remove waste products, they would provide nutrients, and they would defend against infections. So basically they would support neurons in multiple different ways. And so let's go over different types of glial cells that we have. Number one is oligodendrocytes, which will myelinate the central nervous system, like brain and the spinal cord. And two is the Schwann cells, which would again myelinate neurons, but basically it will focus on neurons that are in the peripheral nervous system. The other type of glial cells include microglia, which are basically phagocytic cells that will ingest and break down different debris as well as pathogens. So it will help fight pathogens. The next type of cells are ependymal cells, which line the brain ventricles, and its function is to produce cerebrospinal fluid, which acts as a shock absorber. And the last type of glial cells are astrocytes, which are basically star-shaped glial cells that provide physical support as well as nutrients to the nerve cells, and it also forms the blood-brain barrier in order to control transfer of molecules from the blood into the brain. Next, we are going to discuss the reflex arc, and basically reflex arc refers to our sudden and involuntary response to certain stimuli, like for instance if there is something very painful or irritant, like for instance if we step on a nail or if we touch something extremely hot, we will respond very quickly without even thinking of what had happened, and this refers to the reflex arc. And the way it actually works is that our sensory neurons that are for instance located in the skin will relay information directly to motor neurons with the help of the interneurons. As we just discussed, interneurons, their function was to transfer signals from sensory neurons to motor neurons. So there would be a direct transfer of signals from sensory neurons to motor neurons, as a consequence of which we would be able to right away respond to these painful or irritant stimuli without even thinking about it, because the signal has not yet reached the brain while our muscle found out that it has to make some sort of reaction and retract back. Alright, so basically reflex arc, by not involving the brain, it will have a rapid response to avoid potentially harmful stimuli. Next, we are going to discuss the division of the somatic and autonomic nervous system. So you know how we discussed that the peripheral nervous system divides into motor pathways, and the motor pathways themselves are dividing into somatic and autonomic nervous system. So the first division is the somatic nervous system, which is largely under our voluntary control, and it helps us move our different muscles, like for instance head, trunk, and limbs. The autonomic nervous system, on the other hand, it's not under our voluntary control, and it basically helps control glandular secretions as well as contraction of muscles, like for instance smooth muscles and cardiac cells. Examples of which include regulation of our blood pressure and heart rate by the autonomic nervous system. So basically the system divides into two branches. Number one is called sympathetic nervous system, or fight or flight response, which is mediated by release of the norepinephrine neurotransmitter. And this type of neurotransmitter acts on two types of receptors. It could be alpha adrenergic receptors, which affect smooth muscles of the vessels, as a consequence of which there would be vasoconstriction and increase in blood pressure. Or it could act on our beta adrenergic receptors, like for instance it could act on receptors that are present in our bronchi, or it can affect the heart muscles, as a consequence of which there would be increased heart rate. The other division, on the other hand, is the parasympathetic nervous system, and is called rest or digest response. And the difference between these two systems is that the sympathetic nervous system is being activated once we are under stress, and norepinephrine, by increasing our heart rate and by increasing our blood pressure, is basically preparing us for any potential danger that we are encountering. So that's why we say either fight or flight response. Versus the parasympathetic nervous system, that's when we are under a relaxed state, and that's why it refers to as rest or digest response. And this system functions by release of the acetylcholine neurotransmitter that acts on muscarinic receptors, and so consequently there would be decreased heart rate, increased secretion of exocrine glands, increased peristalsis and bladder contraction. So now we can relax and urinate, and increased peristalsis helps with the digestion of food. And then there would also be pupillary constriction. So here I have summarized the difference between sympathetic and parasympathetic nervous system in terms of their site of action. Regarding the pupils, the parasympathetic nervous system causes constriction of pupils, while the sympathetic nervous system causes dilation of pupils. So think of it, once we are scared, our pupil is dilated because we have to take as much visual input as we can. So that's an example of, again, fight or flight response. Again, SNS causes secretion of viscous saliva, while parasympathetic nervous system, when we are ready to rest and digest, will secrete watery saliva. Other effects include, for instance, decreased heart rate or constriction of bronchi, versus the sympathetic nervous system that increases our heart rate for better pumping of blood and dilate bronchi so that we inhale oxygen better and we are ready to either fight or run away, which is flight. And then the other system is acting on the digestive system, and so you can see how the parasympathetic nervous system stimulates processes that enhance digestion. So there would be increased digestion process of food, versus the sympathetic nervous system will put off these processes because they are not very important at the moment. We have to think of our own survival. So therefore it inhibits stomach motility and secretion. It inhibits gallbladder. It inhibits intestinal motility. Likewise, it will cause relaxation of bladder so we don't have to urinate at the moment, versus the parasympathetic nervous system that induces contraction of bladder so that now we can urinate. And then the other system that is being affected by the sympathetic nervous system is the adrenal glands. So basically the adrenal medulla, which is the center of the adrenal gland, upon stimulation by the sympathetic nervous system, could release the epinephrine and norepinephrine hormones in order to further enhance the effect of the sympathetic nervous system. So this is again an example of integration of the nervous system with the endocrine system, where the nervous system can act on the endocrine system directly. Alright, so that summarizes basically the effects of the autonomic nervous system on different organs. If you need to pause this video for a few minutes to review these systems, you can do that. Otherwise, we are ready to move on to the next topic, which is the structure of the nerve cells. We already know that nerves are specialized cells that help transmit impulse, but in order to do that, they will have to have different structures to receive signals and then transmit them to the next set of, whether they're next set of neurons or next set of effector cells, like for instance glands or muscles. So the first structure of neurons is the dendrites, which is basically branch-like extension that comes off from the cell membrane, and their function is to receive impulses from other neurons or other sensory receptors in the cell body. So dendrites will receive signals. Next we have the cell body or soma, which contains the cell nucleus, mitochondria, and other organelles. Now the signal that is received by dendrites, if it's significant enough and large enough, then the soma or the cell body will relay this signal to the axons. And so next we have axons, which is basically a structure that conduct impulse away from the cell body. And so this one will conduct signal away from cell body or from soma until the signal reaches the axon terminal, at the site which it will release the neurotransmitter chemicals. And it could therefore affect the neighboring cells. And neighboring cells could be, for instance, it could be a gland, it could be a muscle, or it could be another nerve cell. Now, we will discuss later on the functions of the myelin sheath, but as we just discussed, we have two types of glial cells that forms the myelin sheath. And those, could you tell me what they were? For the central nervous system, we had oligodendrocytes, and for the peripheral nervous system, we had Schwann cells. So this is here, you can see that we have the myelin that is formed around the axon, and basically this myelin will provide an insulation so that the signal as it's being transmitted here can pass on quickly by jumping from one point to the other, rather than slowly being transmitted throughout the entire axon. So basically myelin sheath, its function is to speed up the transmission of impulse. So this is an example of a myelinated nerve, And there are also other nerves that are not myelinated, as a consequence of which transmission of signal in those nerves is slower compared to the myelinated nerves. So next, let's talk about how a nerve impulse will be transmitted. And so just to provide some background information, all cells have a membrane potential due to the uneven distribution of ions, like for instance sodium and potassium. And so here you can see in this example there is a high concentration of potassium inside cell. In fact, it's usually 110 millimolar per liter concentration of potassium inside cells versus the concentration of sodium outside the cell, which is about 140 millimolar per liter. And this imbalance is being induced by the potassium sodium ATPase pump. So here you can see that I'm showing you a pump which brings in 2 potassium ions in exchange for 3 sodium ions that are being exported out of the cell. So here is another image of the sodium potassium pump. You can see that we have inside the cell at the bottom of the image and outside the cell on top of the image. So by using ATP, this pump is transferring 3 sodium out of the cell, and in exchange it will bring 2 potassium inside the cell. And consequently, by pumping more positive ions out of the cell than what is being pumped inside, we will have accumulation of plus charge outside the cell, and so therefore the membrane potential inside the cell will be negative. So sodium potassium ATPase pump is one factor that attributes to the negative membrane potential of cells, which is about 60 to 70, I should say minus 60 to minus 70 millivolt. Other factors that attributes to this negative membrane potential includes, like for instance, presence of negatively charged proteins inside the cells. And then finally, molecules in an environment will try to leak from area of high concentration to area of low concentration. And cell membrane does have some permeability, so therefore K plus will leak outside the cell while Na plus will leak inside the cell. However, permeability of cell membrane for potassium is more than sodium, as a consequence of which the amount of positive ions that leak out is more than the amount of positive ions that are leaked inside the cell. And so consequently, there would be increase in negative potential until we reach a potential of minus 60 to minus 70 millivolt. Alright, so we just discussed 3 different mechanisms that would attribute to the negative membrane potential. So here you can see our resting membrane potential inside the axon right here. We have negative inside the cell versus positive outside the cell. Now what happens once there is an action potential? So once there is an action potential, it will cause opening of the sodium channels, as a consequence of which sodium will now enter the cell and cause increase in positive membrane potential. And this action potential will keep on going through by activating more sodium channels and so on. Okay, so that leaves us with the next concept, which is how an action potential is being created. So basically what happens is that once the resting membrane potential of a cell reaches a specific threshold, so here you can see that we have about minus 60 to minus 70 millivolt resting membrane potential. That's what we've been just discussing. Once there is a stimulus that pushes this resting membrane potential, making it a little more positive until it reaches a specific threshold, that's the time that it will cause opening of sodium channels. So now there is opening of sodium channels shown in red, as a consequence of which a depolarization wave will happen, where there would be more positive charge inside the cell compared to outside. And then during the next phase, once the membrane potential increases, now it's time to bring back the membrane potential to its original resting potential. So that's the time that the potassium channels open in order to let potassium outside the cell. And by letting potassium outside the cell, again, we will have more negative inside the cell, and so now resting membrane potential is initiated. So that's another function of the sodium-potassium pump. You see how we have so much sodium outside the cell and so much potassium inside the cell. So with depolarization, what happens is that it will cause opening of sodium channels and sodium will flow in. As a consequence of which, the resting membrane potential all of a sudden increases. And then in the next phase, which is the repolarization phase, the potassium channels open. And so now potassium will come out of the cell, as a consequence of which there would be repolarization. So that's basically what we've just described here. We first described how the cell maintains a resting membrane potential, and then next, by opening the sodium channels, once a certain threshold is met by increase in the resting membrane potential, by opening the sodium channels, there would be increased positive charge inside the cell. And then later on in the next phase, which is repolarization, by opening the potassium channels, potassium will get out of the cell and the cell membrane potential will be restored. Now here I would like to focus on the concept of all or none law of threshold potential. And basically what this means is that in order for depolarization to happen, we have to reach the threshold. It doesn't matter if our stimulus reaches just close to the threshold and then it goes back down, we still won't have depolarization. In order to have all effect, or in order to have the depolarization, we have to reach the threshold. So once the threshold is met, we will have an all response, meaning that depolarization has happened. But if we don't reach the threshold, then nothing will happen. So all or none response. That's basically the concept that we are referring to. Okay, so here depolarization between -70 to -55 millivolt will have no effect, while any depolarization from -55 to 0 millivolt will induce an action potential. Okay? The next concept we've already briefly touched upon is the role of the myelin in increasing the speed of the impulse transmission. So basically myelin provides insulation to axons, and consequently the only places where an action potential can happen is the space between two myelin, which is referred to as nodes of Ranvier. And by providing this insulation, the nerve doesn't have to undergo depolarization at every step. Rather, there would be a jump from one node of Ranvier to another one for inducing depolarization. And this process is called saltatory conduction, which makes the transmission of impulse at a much faster rate. So faster rate of impulse transmission. Okay, so far we've discussed the resting membrane potential, how an action potential is initiated, how myelin helps with a quick transmission of action potential. Next, we would like to talk about how signals are being transmitted across a synapse. And so here I'm showing you the end terminal of one nerve cell, and next we have the receptor of the next cell, which could be another nerve cell, or sometimes called postsynaptic cell. It could be a muscle, or it could be a gland. So basically here I'm just generalizing the mechanism of signal transmission across a synapse. So what happens at this stage is that there would be synaptic vesicles that contain the neurotransmitters. In order for neurotransmitters to get released into the space between the effector cell and the neuron, there has to be influx of calcium. And so once there is an influx of calcium, now the synaptic vesicles can fuse with the cell membrane, as a consequence of which neurotransmitters will be released into the synaptic cleft. So basically synaptic cleft is the space between a nerve cell and the next cell that receives the signals. So from here on, now the neurotransmitters can move on and act on the neurotransmitter receptor that is located on the next cell. All right, so once again, we had influx of calcium that allowed infusion of the neurotransmitter vesicles with the cell membrane, as a consequence of which neurotransmitter were released into the synaptic cleft and would bind their receptors. Now here I just gave you an example of acetylcholine. It could have been norepinephrine if we were talking about sympathetic nervous system, which would act on its norepinephrine receptors. Now here next I would like to discuss different type of the neurotransmitters, and these include acetylcholine. And so acetylcholine, it can be used for transmission of signals for motor neurons of the somatic nervous system, like for instance our skeletal muscles are being activated by the acetylcholine receptor. And we also discussed how the parasympathetic nervous system also gets activated by the acetylcholine receptor in order to induce the rest or digest response, cause vasodilation of our blood vessels, and induce other effects that we discussed earlier in this lecture. Next, we have the norepinephrine neurotransmitter, which is being used by the sympathetic nervous system in order to prepare the body for potential dangerous or stressful conditions, as a consequence of which it will induce a fight or flight response. Dopamine is another important neurotransmitter that can affect both movements of the body as well as feelings of pleasure and other sensations. Therefore, patients who have excess dopamine secretion can have schizophrenia, while Parkinson patients have decreased number of dopaminergic neurons, as a consequence of which they have difficulty with movement. So again, dopamine can regulate body movements as well as sensations of pleasure. Next, we have serotonin neurotransmitters, which regulate mood. Consequently, patients who have low level of serotonin are generally depressed, and in fact, the antidepressant medications are mostly targeted at increasing the serotonin concentration in our body. And then finally, we have endorphin, which acts as a painkiller while affecting brain emotional areas. And that's why that patients, for instance, that are taking morphine, it helps them by killing their pain, but they also enjoy taking morphine. That's why there is a high risk of addiction to these painkillers. The next concept I would like to discuss is what happens after a neurotransmitter is being released into the synaptic cleft. So let's talk about acetylcholine, for instance. Once the neurotransmitter is released here, these neurotransmitters has to be removed at some point. Otherwise, the next cell will keep on getting stimulated and stimulated. So in order to remove the neurotransmitters from the synaptic cleft, there will be some enzymes that will break down those neurotransmitters. And here, for instance, for acetylcholine, the name of the enzymes that breaks down this neurotransmitter is called acetylcholinesterase. Consequently, acetylcholine will now be dissociated into choline and acetate. Now, choline will be transported back into the axon terminal, and the nerve cell can use it to make more acetylcholine neurotransmitters. Okay, so in particular, I wanted to give you an example of acetylcholinesterase because that's the topic that could be asked, like for instance, how different gas nerves can affect the concentration of acetylcholinesterase, as a consequence of which it will increase the stimulation of acetylcholine receptors. All right. Next concept is the difference between excitatory and inhibitory neurons. So with the first type, excitatory neurons, they would activate the postsynaptic cells by increasing the influx of positive charge, like for instance by opening the sodium channels and allowing influx of sodium, as a consequence of which the next cell, the postsynaptic cell, will be activate. So activate postsynaptic cell. Versus the inhibitory neurons, what they do is that they trigger opening of potassium channel, as a consequence of which potassium will leak out of the cell and cause hyperpolarization of the postsynaptic cell. And so therefore these type of neurons are referred to as inhibitory neurons because they prevent any response initiation from the postsynaptic cells. Next, I would like to discuss the summation phenomenon. Summation, which refers to the additive effects of multiple presynaptic signals that reach a postsynaptic nerve. So additive effect of multiple presynaptic impulses. And there are two types of summation effects that we can have. The first one is called temporal summation, where a single neuron sends multiple signals, and that's why it's referred to as temporal, meaning related to time. So this means related to time, in that a single neuron fires multiple signals over a period of time, versus spatial summation, where multiple impulses from multiple presynaptic neurons are being received simultaneously. And that's why spatial means relating to space, because now we are talking about multiple neurons that send signals simultaneously. All right, so that's the difference between temporal versus spatial summation. The next concept to discuss is firing frequency and refractory period. So what happens when a large depolarizing current reaches a nerve cell? It can induce multiple action potential. In fact, the frequency of firing reflects the magnitude of the depolarizing current that has reached that nerve cell. But then there is a limit to the maximum firing frequency, which is 1000 Hertz. And the reason for that is that once an action potential is initiated, the cell needs some time in order to recover and initiate another action potential. So therefore, this is referred to as refractory period, where the cell is limited for initiating the next phase of the action potential. So we have two types of refractory period. The first one is called absolute refractory period, during which time there is absolutely no chance of initiating another action potential. And this is due to the fact that right after depolarization, there would be inactivation of sodium channels. And so therefore, if sodium channels are inactivated, there won't be any depolarization to start an action potential. And these sodium channels will remain inactivated until the cell will regain some of its negative potential. So there would be absolute refractory period until repolarization happens. Now, after the absolute refractory period, this is the time that potassium channels are letting potassium outside the cell. So now in this period, the sodium channels have been activated, and so therefore a new action potential can be generated, but it requires a greater than normal stimulation or depolarization, because now the potassium channels are open, and so therefore a stronger stimulus is needed to reach the threshold potential. So again, we have two types of refractory periods. The first one is called absolute refractory period, when we can absolutely have no new action potential due to inactivation of the sodium channel. Once the cell starts to regain its negative potential, that's the time that the sodium channels will be activated. But then at the same time, since the potassium channels are open, it will take higher than normal stimulus, or I should say stronger than normal stimulus, in order to initiate another action potential. And this is referred to as relative refractory period, meaning that it's possible to initiate an action potential, but a stronger than normal stimulus is needed. And then the final concept I would like to discuss here with you is integration with the endocrine system. So we've already discussed how the sympathetic nervous system, for instance, it will act on the adrenal medulla to induce the release of epinephrine and norepinephrine. And so here we have the nervous system integrated with the endocrine system. Another example that I can give you is, for instance, the hypothalamus that acts on the pituitary gland, and then pituitary gland releases different hormones that can act on adrenal cortex, or it can act like, for instance, the ACTH that acts on the adrenal cortex, or the thyroid stimulating hormone that acts on the thyroid. And again, this is another example that the nervous system is acting on the endocrine system. All right, and an endocrine system can also affect the nervous system. In fact, during embryonic development, for instance, our sex hormones can affect the number of receptors on our central nervous system. And that's the reason that males, for instance, have a stronger response to testosterone versus females that have a stronger response to estrogen. So in any case, there is an interrelationship between endocrine and the nervous system, and these two systems are integrated. All right, so next, I would like to answer some short questions before we move on to the practice questions. So the first question is, what is an axon hillock? And here is a drawing of a nerve cell. And basically, axon hillock is the part of the axon that originates from soma. And thus, this is sometimes referred to as spike initiation zone, because if the summation effects of all the presynaptic impulses depolarize the soma, as a consequence of which there would be enough depolarization of the axon hillock, that's the time that we will have an impulse transmitted down the axon. And here this nerve is an example of an unmyelinated cell. You can see that we don't have any myelins around the axon here. The next question is, what is a synapse? So basically, the connection between a nerve and a postsynaptic cell refers to as synapse, and it could be, for instance, a connection between two nerves. It could be a connection of a nerve with a muscle or a nerve with a gland. And this synapse, as we just shown it in our earlier images, is composed of a nerve terminal. It's composed of a postsynaptic membrane that has receptors, and then it's composed of the space in between that is referred to as synaptic cleft. What is the mechanism of action of local anesthetics? Like, for instance, lidocaine. How does it function? So basically, in order for these anesthetics to function, they have to inhibit initiation of an action potential. And so lidocaine, for instance, it functions by inhibiting the sodium channels. Okay, so if sodium channels are blocked, therefore we won't have any depolarization, similar to what had happened in our absolute refractory period where the sodium channels were inactivated. Okay, so the mechanism of local anesthetics like lidocaine is inhibition of sodium channels. Next question is, what structure lies between the dorsal root ganglion? And basically here I've already shown you the image. It shows you in blue color the sensory nerve cell, then we have interneuron in orange, and then finally we have the green efferent nerve. And so again, this was another example of reflex arc. And inside the dorsal root ganglions, we have the cell bodies of the sensory neurons. So here you can see we have the cell body of sensory neurons. So the answer to this question is cell body or soma of sensory or afferent neurons. The next question is, what are preganglionic neurons? So basically, in the autonomic nervous system, which includes the parasympathetic and the sympathetic nervous system, there are two neurons that are used to transmit a message. And the first neuron that is used to transmit a message is called a preganglionic neuron. And the second neuron that receives signals from dendrites and then via the axon send the signals out, this is called a postganglionic neurons. And all the neurotransmitters that we were talking about was acetylcholine for parasympathetic nervous system versus norepinephrine for the sympathetic nervous system, we were all talking about postganglionic neurons because those are the nerves that will induce the final effect. If we wanted to talk about preganglionic neurons, then I should have mentioned that both the SNS and PNS are releasing acetylcholine, but then it's the final neurotransmitter that matters. With the parasympathetic nervous system, acetylcholine is released in order to induce the rest or digest response, while with the SNS there is norepinephrine that will induce the fight or flight response. Alright, so again, for the preganglionic neurons we have acetylcholine that is being released by both systems, but it's acetylcholine that is being released by postganglionic neurons of the parasympathetic nervous system versus norepinephrine that is released by postganglionic neurons of the sympathetic nervous system. The next question is, what is the difference between monosynaptic and postsynaptic reflex arcs? So with the monosynaptic reflex arcs, the sensory nerve is directly synapsing on the efferent nerve. So here we have the sensory nerve directly synapsing onto the efferent nerve, as a consequence of which, since there is only one synapse, we call it monosynaptic. On the other hand, once we have interneurons in between, that's the time that we call it polysynaptic, because now we have multiple synapses. And here, this example was an example of a polysynaptic reflex arc. The next question is, describe the Nernst equation. And basically this is a general chemistry topic, but since examiners like to relate Nernst equation with the membrane potential in cells, I decided to include this topic here as well. So basically what Nernst equation does is that it will help calculate equilibrium potential across a membrane based on an ion valence as well as its concentration gradient. And so we just discussed how potassium is an important determinant of membrane potential in cells due to the high permeability of potassium. And so here we can use the Nernst equation in order to calculate the membrane potential in cells based on the gradient of the potassium ion. And so with the Z value we have number 1, because potassium has only one plus charge, and then we can incorporate the values for extracellular and intracellular to find out how it will affect the membrane potential. And here let me give you an example. Let's say that just for argument's sake, our extracellular potassium concentration is 1 millimolar versus 10 millimolar for intracellular gradient. Consequently, Ln value of 0.1 would be equal minus 2.3. So that would attribute to the minus charge of our membrane potential. Now in another example, let's say that the gradient further increases and our extracellular concentration is 1 millimolar versus 100 millimolar intracellular concentration of potassium. This would yield us Ln value of 0.01, which is equals minus 4.6. So now there would be further increase in the negative charge of our membrane potential. That's why as the gradient of potassium increases, there would be more negative membrane potential that we have. Okay, next move on to some practice questions. I'm going to read these questions for you. Once I'm done, I will come back and answer these questions. If you need more time, you can of course pause the video. Which of the following cells are required for the maintenance of the blood-brain barrier? Next question. Which of the following phenomenon describes the absolute refractory period? Next question is, amphetamine drugs activate the sympathetic nervous system. What pupillary changes are likely to be observed after using amphetamine? Next we have serine is a nerve gas that inhibits acetylcholinesterase enzyme. What symptoms are likely to be observed in these patients? And then finally, we have how does the vagus nerve affect the heart rate? And basically vagus nerve, I'm giving you a clue, is part of the parasympathetic nervous system. So let's go back to the top. Which of the following cells are required for the maintenance of blood-brain barrier? We just discussed how astrocytes are required for maintaining physical supports, providing nutrients, as well as for the maintenance of the blood-brain barrier. So choice B would be the correct answer. Can you tell me what was the functions of oligodendrocytes? They were myelinating the central nervous system. And then how about Schwann cells? They were myelinating the peripheral nervous system. Microglia, their function was to phagocytose the pathogens. Then we have ependymal cells, which were lining the ventricles of the brains in order to produce cerebrospinal fluid, and its function is to act as a shock absorber for the central nervous system. Which of the following phenomenon describes the absolute refractory period? And the answer is inactivation of sodium channels. It's not closed sodium channels because once we are not having any depolarization and once the cell is in a resting state, we do have closed sodium channels. It doesn't mean that they cannot open. So inactive sodium channels is what leads to the absolute refractory period. And then if we were asking which of the following describes the relative refractory period, the answer would have been opened potassium channels, because now the cell is undergoing repolarization, and so a stronger stimulus would have been needed to induce depolarization and activation of the action potential. Next question is, amphetamine is part of the sympathetic nervous system. What pupillary changes are we going to see? So referring back to the table, or let's say the figure that I've provided you here, you can see that the sympathetic nervous system releases norepinephrine in order to cause dilation of the pupil. So the answer for this question would be it will cause pupillary dilation due to the release of norepinephrine. If we were talking about parasympathetic nervous system, on the other hand, the answer would have been it would cause pupillary constriction due to the release of acetylcholine. The next question is, serine is a nerve gas that inhibits acetylcholinesterase. What symptoms are likely to be observed in these patients? So referring back again to the figure with acetylcholinesterase, if serine inhibits acetylcholinesterase, what happens next? There would be increased concentration of acetylcholine in the synaptic cleft. So therefore, there would be increased activation of the parasympathetic nervous system. And so therefore, all the symptoms that here we have described in the figure right here would be exaggerated. So all these symptoms would be exaggerated by application of the serine gas. And so let's see which of the answer choices describes a parasympathetic nervous system response. Pupillary constriction, that would be correct. And then we have urinary retention, which is a feature of the sympathetic nervous system. So is constipation. If you recall, parasympathetic nervous system was activating the detrusor muscle in order to initiate urination, and it would also cause peristalsis of a GI tract to enhance digestion. And so none of these answer choices would be related to PNS activation. And then finally, PNS also enhances secretion of watery saliva, and so therefore dry mouth and nose would not be a feature of PNS. In fact, that would be a feature of the sympathetic nervous system. The last question is, how does the vagus nerve affect the heart rate? And so vagus nerve is part of the parasympathetic nervous system. It releases acetylcholine, which causes decreased heart rate. So the answer would be reduced heart rate by increasing acetylcholine. Now, if we were asking about the sympathetic nervous system, under that circumstance, the answer would have been increased heart rate by releasing norepinephrine. All right, so that concludes our discussion of the nervous system. Thank you all for listening, and we will see you for the next lecture.